So we're going to begin, and we've begun. Housekeeping materials. I sent you, I basically finalized the exam. So I sent you this, whatever this outlining thing is. Edit, delete, download. So it wasn't a whole lot. That was the same one I just did. Wherever that. That's a supplemental. No, I downloaded that twice. Ignore me. I'm trying. I did the wrong thing. Well, the additional study guide, that thing. I'll mess it up if I try to download it. Nevertheless, there's just a little bit from the start of cells because I wanted to really set the frame, bring proteins into it and what they do in the membrane, and then really explain a little bit about some of the early uses of proteins so that when we start talking about that, even if the material's not on the test, it kind of reinforces it with you. So as we start to talk about the next topic after the test material, after we identify the membrane proteins and how cells are latched together, at that point we start talking about a thing called movement across membrane. So while I talk to you about that, even though it's not on the exam per se, it'll reinforce the roles of those proteins, so it should help, hopefully, to cement that a little bit better in your minds. Okay, that's kind of the method to the madness, so to speak. So, but the only problem I had with some of these PowerPoints are large, particularly because not so much from the Welsh Hull textbook. When I use the Marib textbook, there are some large PowerPoints. One of them I just sent to you because I have to send as an attachment, an email, and I think it has to do with animations that they have a lot of, which I love. Because I really feel they help. So look out for those. And I put a little note that I had to send it to you. And I tried to upload it here, and they went, "Oh, you can only download it. You can't upload it." So they switched to Ultra. This Blackboard Ultra, whether you've used Blackboard before, has a lot to do with accessibility and ADA, Americans with Disability Act requirements, and stuff like that. So there's some good and there's some bad. For me, it's I don't use all the features, so for me it's just sharing documents with you, as opposed to having to print them constantly like we used to do those many years ago. Voila! And the last topic, thankfully, in biochemistry is nucleic acids. This is the other one that we see an abundance of nitrogen, and as you will see, phosphates as well. So those are the big players when it comes to nucleic acids. Yes, and so they're the polymer. The building blocks are different, similar to proteins in the fact that they're like amino acids, they vary by the R group. Here, there's only really 4 that are used in RNA and in DNA. Even though they're not necessarily identical, there's one flip or one change. And so nucleic acids are the polymer, and we call the monomer are called nucleotides, and they all have the same composition. They have a nitrogenous base. Why am I making a point about nitrogen? Again, I commented on it a little bit when we were talking about proteins. We've got to be trouble with nitrogen. It's hard to dispose of. Our kidneys they're almost job one for the kidney is nitrogenous waste removal. So things that containing nitrogen, we only have so much running around in our system. And our kidneys, it's a challenge to get rid of that stuff. That's why I'm making a point about it. So what happens, these nucleotides have a base containing nitrogen, and you're going to see it has carbon and nitrogen bonded in these sort of interesting arrangements that you will see. It's either going to be a single ring or a double ring. You do not have to know anything about the structure. You do need to know whether it's a single ring or a double ring, and they have a classification name that you'll see as well that you need to be familiar with. The sugars are straightforward. 5 carbon sugars, and I showed you them a couple of times already. The difference is one's missing an oxygen in the same position. So you get ribonucleic acid versus deoxyribonucleic. Deoxy without an oxygen. And you have a phosphate. So what's DNA? The magic. It is, what's interesting about this is the dual function that has is extraordinarily vital. It's certainly the most remarkable, I think, of all the molecules when we think of it, particularly the macromolecules, because it includes the entire genetic group blueprint And I was looking a little bit more for some of the stuff I told you. We even have more similarities. If you really want to look at the genetic things, we're almost about less than 1% really counting for the substantial differences that we visualize, more visible that we see with each other. I'm not talking so much about genetic diseases and things like that are there. And it has this miraculous molecule that they spent forever trying to figure out. And I touched on it a little bit earlier. This is historically. Scientists knew after World War I, before the start of World War II, they were very interested in what is DNA, because we see it in all the cells, and we got an important feeling that this is really telling the cell what to do. And we know it basically makes all the proteins and then replicates. How does it do this stuff? All they knew were the compositions. They knew consistently they saw certain relationships between the molecules. They couldn't figure it out. They knew, and you're probably familiar with this, this is what they knew. Okay, you have 4 basic molecules. You're going to see the structures. Adenine and guanine are called purines, just the name to know. And the purines have a double ring. So it's 2 carbon-nitrogen rings. I can't draw the diagram. It's sort of like this. And then pyrimidine, right? Pyrimidine. Have a single ring. Like something like that. Nevertheless, and they knew the following things. They knew that they could identify the structures, they separated them chemically. But what they knew, even though we all just use the letters, the A's and the G's, so I refer to adenine and guanine, and cytosine and thymine were always there. But what they found was a fixed relationship. They always had an equal number, molecule-wise, of adenines and thymines. So the A's and T's were always equal, and the cytosines and the guanines. Now, maybe retrospectively, you might be able to figure out, now since we know they found out that the structure was a double helix, retrospectively, if you knew that going in, you probably could have figured out it's stronger. But they basically, after what happened was when World War II was about to start, all the scientists stopped looking at something in research and started to do stuff that was related to the war effort. So this interest in DNA lagged until the conclusion of World War II. That's why the history is kind of important. And it was around 1948 or 49 in England where Watson and Crick and another researcher, and eventually in 53 they were awarded the Nobel Prize. They couldn't figure it. They knew the components. They had a pretty good idea what it was like. And I told you the story. This was a lady who worked with them named Rosalind Franklin, who never got credit, typical of science and medicine then, and to a certain extent now, which is terribly wrong. And she did X-ray diffraction, meaning she took the DNA and they were taking X-rays of it. And it was never clear that they couldn't see the shape. And it came to her, how do I slow it down? Well, I'll freeze it. And we're not just talking about putting it in the freezer. She's talking like liquid nitrogen freezing it, making it so it would be as frozen as possible, ergo as least motion as possible. She began to measure. She saw this twisted ladder formation, and she saw the rungs were all the same in the sense of their width. And it figured out that it was equal to a 2 and a 1. So you were always going to have, they would bond together. So you had basically a 2 and a 1 here, and a 2 and a 1 here, always. And it's magic. And the story is told that she went down having done that in the morning, and everybody there, wherever they were having lunch in whatever laboratory or facility they were working at, she said, I just did this measurement. It's incredible. This is the exact measurement, and it's equal to 1 pyrimidine and 1 purine diameter for these rungs, and it appears to be a twisted ladder. And they went up and built the molecule and even though she gave them the critical piece, never mentioned her name until she can't receive the award posthumously. She received recognition posthumously. That's interesting. Sometimes Nobel Awards are only given to people who are alive unless it has been awarded and they die before the ceremony, then they'll still receive the award. Just, it's a curious thing about the Nobel. I like this stuff. It's okay. You didn't know this is part history class. So DNA holds the genetic blueprint for the synthesis of proteins. We knew that. And plus, this is how we make new cells and how the cells are identical if we want them to be. And the vast, vast majority of the cells we make are called mitosis, which you're already familiar with. Asexual reproduction is all about making them the same every single darn time. Okay, so that's the whole idea. And she was the one who came up with the idea that we call it complementary base pairing. A always pairs with T, G always pairs with C. Simple. And this is the structure. So that's what it was those, even though it isn't as easily demonstrated in this graphic, okay, that again, G's and C's, A's and T's. And what happens if they're not paired exactly, which does happen with some degree of frequency in life. Okay, so if this is an A and a T and you're a G and a C, and all of a sudden I go back over here, I got an A and an A. We have a name for that. Mutation. Error in DNA. You'll see down the road, we have a lot of mechanisms in our cells that other cells don't necessarily have for taking care of mutations. So mutation doesn't mean that it's something awful. Oh my God, I'm going to die. I'm going to have an extra eye in the middle of my forehead or something bizarre. Like that's not. It means there's an error in DNA. That's all it means. A lot of times they don't mean anything because it depends on where they fall in the strand or in something called the genetic code, which we're not going to do in this class. Someone else wants to do it with you, we've had it done before, that's fine. So mutations are, surprisingly, we make a lot of mutations, very few of them impact us. Sort of we make billions of cells every day. You're going to mess up having a few mutations. Most of the time, it doesn't make a damn bit of difference. Your body gobbles them up anyways. It undergoes a process you may have heard of called apoptosis. Ringing a bell? Called programmed cell death. APOPTOSIS. We'll get to it in the cell cycle. And it basically is just like a suicide switch for the cell if things are not assembled properly. Forget it. We've got a problem. So here we are. This is what it looks like. You have consistently, you have a phosphate group. In this case, there's a whole difference, that absence of that oxygen there, making deoxyribosis allows this shift. I have one tiny loss of one atom to change DNA and RNA. It's remarkable things that they can both do realistically. And you have it, and you'll notice one of these, just looking at it, side up, the other upside down. We have a fancy word for this. Okay, it's called anti-parallel. They're identical segments. That's the beauty of DNA. When it opens and closes, it doesn't matter whether you start at the bottom or start at the top. We're doing it concurrently. We're replicating the DNA in both directions. One is up and one's down, but they're identical strands. That's very significant as well. So it doesn't matter where, once you open it. It's a little bit different how they're assembled, and we're not going to do that. There's stuff called leading strands and lagging strands. She's doing that in the other room, in genetics. She's doing that. Love my paper. Nevertheless, you see what's holding them together. Hydrogen bonds. It's a weak magnetic bond. Every time so it opens and closes. DNA is a biological zipper. That's the best way to understand what it is. So we call this phosphates and the sugar backbone. Not only are they bonded to each other, bonded to the next one in line. So we have, now just put a T and an S base, and a T and an S and a base like that. And on the other side, you have a base, a sugar and a phosphate, and a base, a sugar and a phosphate. Even though they're going in opposite directions like this, effectively what happens, okay, phosphates and the sugars bond with each other as well. So what that means, we call that the backbone of this molecule. So the only variation has to do in the area where the bases are. As you can see, here you have the classic adenine and thymine. I'm making a big deal about it down the road. Many of you, when you get into things like pharmacology or physiology or pathophysiology, better for those going into clinical fields, you learn it just becomes a lot more relevant. A lot of the way we treat problems are with medicines that utilize this. We make agents that look a lot like this, have chemical substitutions that are really poisons or will not let those strands form. That's particularly used in antiviral therapy and in cancer chemotherapy. If you know somebody has a cold sore and they took a Valtrex, that's how it works. It's substituted here for an admin. See, that's why you may wonder, and I know you must leave this room, why that? Why in heaven's name is he thought I was going to say a bad word. See, if I was me, I go, WTF is he talking about this stuff to me? I don't give an FF. Flying. See, we're careful. This is why. You'll like me now. Wait till the test. You'll hate me. Say bad things about me, like you say about Dr. Robbins, who's only on the short list of nicest guys in the world. Okay, that's it. That's the whole big deal about this. Adenines and thymines bond together. Cytosines and guanines bond together. It doesn't matter. They're like this. You'll see more of the illustrations like this. There's a lot of that stuff. I don't really have a good animation, but it consistently forms when they say it's called an alpha double helix. It's a spiral staircase. With the weakness of the hydrogen bonds, it opens and closes. Voila. The difference. Put back the oxygen, it's about the shape. It can't assume that shape. Rather than being a double molecule that's spiral, we have a linear molecule. The advantage to that being linear, okay, DNA is so large, it can't escape the nucleus. RNA can. That's why RNA is what effectively sends the message to an area called the ribosomes to make proteins. RNA is a big player. Okay, it's DNA is the largest molecule. RNA is a little bit different. It is single-stranded. It is active mostly outside of the nucleus, even though it is made effectively all in the nucleus. There's little things when we get to the nucleus called nuclear pores, which you may have heard of, that allow stuff to come in as building blocks and allow RNA to go out. So DNA is stuck in there, but it somehow is able to communicate its instructions. You and I might text somebody or call somebody. They have to do it molecularly. Okay, so the sugar is ribose. Thymine is replaced by uracil. They look identical, as you will see. If they show you the molecules themselves, there are subtle differences between them. Uracil is also in that family of amines. U for it. So you have all of those names that are there. Uracil, and then the cytosine, and thymine. And even down the road, if you have to study genetic code, I'm talking about genetic code, we're looking at it as the code in RNA. So wherever there would be a T, we replace it with a U. And there are 3 varieties. We're going to make it simple. Okay. There's as we get into the subtleties, we'll do with it first. DNA, we're not even talking about replicating. We're just going to talk about assembling proteins. So you have the 2 strands, and one of them, and they pull up, it doesn't matter which strand, that they use as what we call the template, the model. Effectively, since they're the same, they're just going in different directions. They're all going to make the same kind of RNA. The one that they make that basically takes it from outside of the nucleus to the ribosome where it can create proteins is called messenger RNA. Without going crazy, you don't have to know this just yet. Things like this, I talk about it more when we do micro. If you're going to be with me in micro, which you don't want to be, okay. It's called pre-messenger. We basically process the messenger RNA. That's kind of one of the ways we get rid of a lot of those mutations that occur. But messenger RNA effectively is sending the message, which is in the code of A. It's got 4 letters for code, and it's how they're put together, those letters. And they have little 3-letter units. That's the basis of what's called the genetic code. And basically, it's basically the instruction manual of how to assemble the protein. So the first process, and these are important names to remember. When DNA writes messenger RNA, it's called transcription. I do not know how they do it now in a hospital. I haven't operated in a hospital in 25 years. So what we used to do is they would either have a dictating machine or a telephone that we would use. How they do it today, I don't know. When we finished an operation, we would do what's called, you had to immediately do your operative report. That means, and it's like automatic pilot for surgeons. Following an induction and maintenance of general anesthesia, the patient was prepped in the usual sterile manner. A curvilinear incision was effected over, blah, 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 that. So it's like mechanics, and you describe exactly what you did. So that would go to someone who would listen to it and type. That is a transcriptionist. How they do it today with voice recognition and that type of stuff, I do not know. I'm sure it has advanced dramatically over the last quarter of a century. I'm sure it has. So that's what a transcription is. In the old days, before we had that, whoever was dictating in the office, they had a secretary, someone who would write it in shorthand and then type it. My mom did that. So she could take shorthand as fast as you could speak. She could type 125 words to stay in copy. Yeah. Professional secretary. Go figure. So, I'm not that good of a typist. Not even close. Not bad. So thymine is replaced with uracil. So the messenger RNA carries the message. Already, other RNA is in the cytoplasm. It's called transfer RNA. And what that does is it's going to pick out the amino acids that's in the cytoplasm and bring it to be assembled at something called a ribosome, which is made of this ribosomal RNA and proteins. Biologically, inside the cell, we need a platform to do anything. Whether we're assembling or disassembling something, whether we're making proteins, whether we're gobbling up nutrients, we're getting rid of waste products, you always need an area where that can occur. That's the busyness of the cell. So for the time being, messenger RNA is receiving the transcribed information, bringing it to the ribosomal RNA in the ribosome, and with the assistance of transfer RNA, is assembling the proteins. That's basically what it's all about. We call that process, when you assemble the proteins, translation. So it took that message, the transcription, the exact message, and translated it into the language of making proteins. Take amino acid 1, amino acid 3, another 1, a number 6, a number 5, chain them together, voila, we got ourselves whatever kind of protein we're making. So I know it's highly complicated, but for us, I want you to be, I will tell you how I phrase one of the questions on the test. I'm doing this, I don't like them. Okay, here's the answer key. Okay, in DNA only, in RNA only, in both D and RNA, neither DNA or RNA. I'll give you a whole bunch of components like adenine and uracil and phosphates and ribose, and you've got to match them up. Okay, be prepared, be very prepared. Not be afraid, be very afraid. The way to avoid test-taking anxiety is to prepare and be confident. You should start working together or individually, whatever floats your boat. Some people like to study individually, some people like to study in groups. Whatever works for you. That's why, and I know this is a novel concept, the only people who email me are, I'm sick. It's okay. I'm happy to do that, and I respect that you're being professional and keeping me in the loop. If you have a question about something we discussed, you can actually email me. You can actually, I know everybody's running into Dr. Robbins' office. I'm getting envious. He has a much nicer looking office than I do. Okay? He's got neat stuff in the office. He's got the big iMac in the office. All I have is old candy wrappers and a bunch of papers stuck in it. It looks like, my wife will tell you, it looks like my workroom at house. Clutter. We found yesterday, are you ready? A Monopoly Sephora edition. You know, the Sephora, if I'm met, did you know there was a Monopoly game for them? That will make a neat gift for somebody. Who knew? Just say it. No. No. Okay, fine. You insist. No.
Polymers made of subunits called nucleotides. One difference between DNA and RNA is the type of sugar their nucleotides contain. DNA contains the sugar deoxyribose, while RNA contains the sugar ribose. Ribose has one more oxygen atom than deoxyribose. DNA and RNA are each composed of 4 different nucleotides which differ in their nitrogenous bases. 3 of the 4 bases are the same in DNA and RNA: adenine, guanine, and cytosine. The 4th base in DNA is thymine. In RNA, it is uracil. The nitrogenous bases guanine and adenine
They look almost identical.
linked rings of atoms. They are called
Just here.
Cytosine, thymine, and uracil each have a single ring, and these 3 bases are called pyrimidines. For convenience, the carbon atoms in a nucleotide sugar are numbered, beginning with the carbon atom bonded to the nitrogen.
Don't have to know the numbers.
Moving around the ring, up to the carbon that is.
Save the numbers. But this is what, down the road, because this is a little bit more than we need, we go in directions. There's a,
3' position.
it's called 3' and 5'. You don't have to know anything about this. Okay. She's teaching that stuff. That's cell and molecular biology. And then and they assemble it. And this is what I mean by anti-parallel. Okay, where RNA is single. Okay. And so it's very interesting down the road. This is, when we get a little bit, those of you who take micro, you'll learn a little bit about ways that the magic of micro from bacteria, we got enzymes that could reliably chop up DNA into specific fragments. That's the basis of paternity testing. If you do Ancestry or 23andMe, that type of stuff. If you're curious, I'm not. With my 3 adopted, my middle one was curious, and he found, and I think he talked with his biological father. Below these many years, this is 5-7 years ago. And he's 41. So, but with that, if the cells and some relatives and stuff like that, I don't care. I was stuck with him for the first 30. Here, take him. I don't need that. But a lot of people are. Some people are. My first wife would be very uncomfortable with that, by the way. So that's the big distinction between the two of them. Not a whole lot else that you have to know for now. What I showed you before is, again, we use those molecules and we had things. If you go over in that periodic table again, we talked about halogens: fluorine, chlorine, bromine. Halogens are basically toxin. Okay, when we add that to something like uracil, that was one of the early treatments for breast cancer, for 5-fluorouracil. They use that 1 through 6, that numbering system, which you don't have to know. Okay. I didn't know any of this stuff. I got a doctorate without knowing it. This was all new. The age of molecular biology was evolving in the late 60s and early 70s. It was not something we needed to know to get a doctorate, to treat people. So it's remarkable. Now we teach it. Some teach it. We'll teach it in universities and colleges all the time. Maybe even some high schools in advanced classes. Possibly. This is the whole structure. Okay. The big deal. So the building blocks are called nucleotides. Versus and those are the monomers. And the nucleic acids are there. Pulled out. Assembling together. At the very least, we take adenine. I'll just draw the adenine. That's what they're doing here. So you need a base. To make a nucleotide, you need a base. In this case, the sugar is ribose. We need a pentose sugar. And we need a phosphate, PO4 negative sugar. That's how you make nucleotide. Nucleotides are not, that may look like a lot of stuff, it is still considered a small molecule. DNA, RNA, or DNA, the biggest by far. Why I'm focusing on that? The single most, and I mentioned it before, the molecule we make the most and break down the most every single day of our lives is that. ATP, adenosine triphosphate. So it's all chemical reactions, immediately energy. If we had extra, immediately go to fat after about a minute. Adenine containing RNA nucleotide ribose with additional phosphate. That in a second. Why we call it and have it on the graphic, when you put an adenine and ribose together, it's got a fancy name, adenosine. Biologically, that's not where this all ends. Okay? You're going to see we're not going to have just one phosphate. We're going to add two more. About adding those two more changes the bond substantially to the point we get something called a high energy bond. So in science parlance, really speaking, if you had guanine and had a ribose with it, you have something called guanosine. And indeed, the reason I'm mentioning that down the road, we can hear a little about that, a little bit about that. But adenosine is so important. That is actually a chemical. So when we make ATP and we start to break it apart, we use the components. So adenosine is a component that plays a vital role in helping us relax, helping us sleep, plays a vital role in pregnancy. And I don't know about you folks, what you do dietarily speaking, but every morning I block adenosine receptors vigorously to keep me awake. Caffeine. So caffeine specifically blocks receptors in your brain and in other areas for adenosine, and that's why all those energy drinks and stuff like that work that way. So I personally prefer coffee, but I used to be a Pepsi in the morning, but and every so often that will rear its ugly head. It's okay. So here you are. Adenine and ribose make adenosine better than I could illustrate it. Here are the phosphate groups. That little squiggle over there is a high energy bond. Why is it high energy? Phosphates have a negative charge. This is how you draw them. PO4 with a negative charge. If you take kitchen magnets, best analogy I can think of, and try to, we all did this as kids. You had those magnets, and we did have ones that come together, and if they had the same charge and you pushed them, they wouldn't come together. You'd hold them and release them, right? I am not the only one who did this in this room. Okay, so imagine if I had not just two, would give you some energy. If you had a third one, it would be very difficult to hold them in place. That's what makes it a high energy bond. We are imparting extra energy to that molecule in order to hold it together. So we have one, and you're going to see we use all of these. The adenosine, I just explained to you. Adenosine with one phosphate, even though it's just a basic nucleotide, plays an important role as a mediator inside certain cellular reactions. You'll get to it. Adenosine diphosphate and triphosphate are constantly exchanging plates. When we need to release the energy, we lop off the phosphate. Don't have to do much. It's ready to go anyway. Okay, they're negative and negative. They're pushing together. Boom. It comes off. That energy can do work when we want it. And then if we have extra of it, it can last for about a minute before we have to put in some storage form, typically in the form of fat. So that terminal phosphate can be transferred to other compounds that can use energy stored in the phosphate bond to do work. Loss of the phosphate goes from, and we use the acronym ATP triphosphate to ADP di or 2-phosphate. And then ADP, if we lose another one, AMP. It'd be nice if they put that all together on the PowerPoint, but they don't. So what happens? Everybody knows. You may wonder, okay, glucose, our basic energy source. We can't just magically create that into ATP. Remember I told you that all biological reactions require an input of energy. Once that glucose enters our cell, the very first thing we do is we take an ATP and we spend a phosphate and put a phosphate on glucose. It's got a fancy name, you don't have to know, called G6P. Glucose in the 6th position of phosphate. And effectively, we have energized it. What we effectively have done is made it more water soluble. That's why there's always, when there's glucose enters the cell, it doesn't just stop. It doesn't reach what we call an equilibrium and stop moving. Glucose immediately is changed on entering in the cell into G6P, and the fact that we put a phosphate on it, it's not glucose anymore. So there's always more outside the cell than inside the cell, even if the glucose is pouring in. It's a very clever thing that our body does metabolically in order to ensure that once we open the door for glucose, it's coming in. That's insulin's job. Very interesting. That's the biochemistry of it that thankfully you don't have to know, but I'm not telling it to you to show off. She knows 1,000 times more than I do. She's much smarter. So voila, that's just showing the reaction. P with a little I means inorganic phosphate because it's not an organic compound. You're going to see some of this again when we start talking about how muscles work. And so in the presence of water, we hydrolyze it, break it apart, and we pull out of water and we pull it back together. So much we do. If you added up all these molecules that we went from this to this during the course of a day, it would equal to approximately your weight. A lot. And so this is some of the thing, and this is where we get into the part where it dovetails into my talking about the cells, which is the start of the next chapter, how we can do work with it. Okay, a lot of times when glucose, okay, is different because there's always more glucose outside the cell. We're going to talk about a thing called concentration gradient. When you have to push something out of the cell and there's always stuff trying to come in, it takes extra energy. It's like you're in a crowd and you're trying to work your way through that crowd to get someplace. It's not easy. So we spend energy to do that. So whenever you see that ATP sign, it means something that we'll get to called active transport. And I'll go over that again. Contracting a muscle. Smooth muscles are kind of neat. When we talk about them, they kind of spiral in shape. And when we energize them, they kind of get kind of shorter and fatter. And sometimes we have to use energy to put two things together. And that concludes that second chapter. So there's be a lot more about going through with that over time. What I sent you previously to sort of bring this into why I presented it the way I did. And this is the one I sent you, but I can't open it up. So if you're on a laptop or something, you can open your laptop and follow along with it. Or just go with it and just look at the thing. You're under no obligation to download anything. But I went to all of this and talked about cell theory. This is if you got a chance to listen to it. And the neat shapes of all the cells. There's a myriad. They said there's 250 different ones. I never bothered to count. But and shape really is a critical piece. When you get into the cell, and we will talk about that a little. I think that's more anatomy. When we begin to look at it in the context of muscles and nerves, we'll talk about these particularly unique shapes here and here. Okay? And the same things most people are familiar with. You have to have a boundary. And they talk about, and that's why we spend some time. We're going to look at the nature of the boundary. We look at the nature of the cytoplasm. We know about the nucleus being in control. So I went over all this. I guess the point that I made was that, it's a very good view of the complexity of a cell. Not all cells look that way. When you get into cytology, looking at cells under the microscope, or looking at tissues under the microscope, we look at the individual cells, and we can ascertain a little bit from their arrangement and things that they do. That's not, I think that, and I don't know, were you looking at microscopic slides with Dr. Robbins? Yeah, okay. I don't know how much he's going to test you about that or not. That's his call. I would basically, some things to me, bone and blood are fairly easy to see. Some of the connective tissues and lining tissues are fairly easy to see. But when we look inside the cell, we're even looking at the relationship, and I commented on this when I did the presentation, the amount of, this is called endoplasmic reticulum. And we'll get into talking about this. It's a fancy thing called the endomembrane system. The membranes are compartmentalized. And so to protect things that we're trying to make, like proteins or other materials, we basically encase them in these membranes. And the amount of each kind of membrane gives us a clue as to the function of the cell. So we encase them in the membrane to protect them. The inside of the cell is nasty. It's got acids and bases and chemicals and stuff going on all the time. And so we encase it. It's like we're protecting it. Okay, as we walk it through from where it's made here until either it's outside of the cell, maybe it gets stuck in here, maybe it gets stuck in here, maybe it gets stuck in the membrane. There's all sorts of different things. And we'll look at that. And I commented on this distinction between, and this is, stuff's either inside the cell or outside the cell. So we have intracellular fluid and we have extracellular for outside. So all that, all your cells on their exterior have some kind of fluid around them. Your blood has a fluid that you'll come to know is called plasma. That's everything but the red cells and the white cells and the platelets. Cerebrospinal fluid, very similar, but different. Joint fluid, similar, but different. Joint fluid is more of a lubricating fluid. It's got a little more proteins that give it sort of a certain degree of viscosity, whereas cerebrospinal fluid, not nearly as much. But cerebrospinal fluid doesn't have any white blood cells. If it has white blood cells in it, it's a problem. That's one of the ways you would diagnose meningitis. I mean, this is just why the stuff is so relevant that's there. And things like that. Saliva and mucus. Saliva's got some protein in it. Mucus has a lot, makes it sticky. And again, all the things you learned about the extracellular matrix with connective tissues, and they have intra and extracellular fluid. And basically they're similar, but not identical. And the whole ball game is that membrane controlling flow inside. Magically, the whole idea when I talked about phospholipids, magically, when we have phospholipids, they form a bilayer when you put them in solution. We don't have to worry about it. It happens automatically. Good for us. Okay, membrane proteins. That's why I did the whole jazz about the proteins, all that folding, the globules. You want me to get the pieces of paper out again? Probably not. I used to do fruit by the foot for that, but I always ate it. It was a problem. I'm a candy addict. We use candy examples. You know why? What's holidays coming up that means candy? That's right, Halloween. If you can't make an example out of candy, fruit by the foot is useful. Wait till we get to those little white papers with the dots stuck on them. Reese's peanut butter cups. I'm having a moment. So what happens, they naturally form, proteins are able to embed, yet move around. Fluid, mosaic model. The cholesterol stabilizes the membrane to allow it to do those things. It acts as a platform. They've got a fancy name you don't have to know called lipid rafts. Little rafts floating around in this magical fatty environment that are platforms. Again, the term platform for that. They're coated in sugar. How about that? Okay, and we're going to talk about different sugar coatings when we get to certain cells, particularly in micro, we have a lot of sugar coatings versus a little bit of sugar coatings. So, I mean, all types of cells have that. When we talk about bacteria, okay, we call it a glycocalyx. Glyco for sugar, calyx for shell. Okay, and they have these cell junctions. That's the very last thing that we go over once I do what's going on inside the membrane itself and the membrane proteins, and that'll be the last part for the first exam. And this is really useful to review. I commented on it, and I mean, to a certain extent, I'm running out of time here to be able to do it. But what you can see are different kinds of proteins. Here, the little bit of yellow, that's the cholesterol. The phospholipid bilayer is self-explanatory. It's hard to see, but you can increase the size of this and look at it differently on your own screens when you review it. We call this a channel protein. Okay. If the channel implies it's always open, it's mostly for water. Another name we use for it is a pore, P-O-R-E, just like you might have pores for perspiration, or aquapores. It's typically water will go through there. Most, not all, the vast majority of cells are permeable, meaning water can flow in and out. Water flows freely. It's an experiment that you probably did. You may have done something called dialysis bags, or maybe using potatoes as osmometers in movement of water in high school. No? No potatoes? Oh. In cooking, when you make, like at McDonald's, we have really good French fries. They double fry them. What they do when they put them through the machine to slice them, they put them in water immediately. It stops them from turning brown. That's good. They don't oxidize. The thing is, if water will go into those potatoes, they get stiff. And then when you fry the first fry is low, it's called blanching. It cooks them through. And the second is about 310. And the second fry is about 375. It browns them. So if you go to a high-end restaurant and they do steak frites, steak with fries, French. Okay, that's how they'll do the fries like that. I know a bit about French fries. Okay. So that is a pour for water. We rely on that. Proteins can be embedded. Proteins can be outside, on the outside. We have extra proteins on the inside. They all have these, and you look, these are short chain sugars. We'll see the roles they play for identification, for things to attach to them. That's what we're leading to. And that was part of the lecture that had all the different things you can look at, and I can't read to you. And so I'll conclude. We'll stop it here. I'll review this next time. And I'm sure we'll be done on Friday with all the material for the exam. It's really the role of the membrane proteins that are there. And I don't do a lot of these animations. And the very part that we end with, if I can get through this, which I probably can't. The very part we end with is when we look, as we finish this idea about these different cell junctions. And that leads us to movement across membranes, which is not an exam. So thank you. Great day. Have fun. So many times. Done.